you can't really give me pushback for just saying the facts. I mean, you might want to, but this is your own literature. So my
Yeah.
strategy is I'm just going to quote your own literature.
Welcome to Real Science Radio. I'm Fred Williams, and today I'm flying solo. Well, not completely. I've got Sal Cordova in studio. What a pleasure that is. Anyways, my co-host Doug, he's on assignment, but we've got a great show lined up. I can't believe I have Sal here. He's traveling from Washington back to, from Seattle
From
back to
Washington to Washington.
Yeah,
Seattle,
Washington to Washington.
back to Washington, D.C.
So I know a lot of our listeners are familiar with Sal Cordova. We've had what I think are some of our best shows on Real Science Radio. And he's just on the cutting edge of a lot of the latest creation science. And he was just at an evolutionary conference, right? A conference on evolution.
It is the #1 conference, to my understanding. It is organized by the Society for the Study of Evolution, the American Society of Naturalists, and the Society of Systematic Biologists. So this is kind of the annual pilgrimage to Mecca, so to speak. And on their official website, you can even see stained glass windows on their banner as if it's a church event. And so I got to be there. It's a long story how I got to be there, but it's probably not as interesting as some of the, what I tried to bring to the table. I was in fear of being canceled from day one. I mean, if someone just Googles who I am, they could figure out who I am. I could have been canceled, not even admitted to the conference. I was scheduled to present. I was first in line on their session on population genetics theory.
So you were first up.
I was first up.
All right.
And you'll see my page. You'll see my handsome face on their official YouTube channel. When you look for the most popular videos for what we call the concurrent sessions, mine was #1.
You were #1.
I was #1 for the year 2025. Now, for all time, it's a different one that's #1 if you want me to mention which one.
Oh, man. Yeah. So you mentioned to it, off air, and yeah, we might as well. So
yes,
it's not surprising.
So for the #1 attraction for all the years that they've been organizing this was from 2022. It was a drag queen. That was a big event, and it is the most watched of their, of the events on their official YouTube channel. But for 2025, yours truly, a creationist, was #1 on their YouTube channel. I got an official letter from them. And I'll read it here. It said, Salvador Cordova, your participation and expertise made the conference a great experience for attendees, and we hope to see you again in future years.
So this is after the conference.
This is after the conference.
Okay.
So I think this is an act of God. They could have canceled me. There are probably some people in that conference that hated my guts. And so I do want to credit the organizers for really, they rolled out the red carpet and said, let's hear views. We're here to talk science, and science invites criticism of even theories that are orthodox, and they were true to the scientific enterprise. So I do want to thank the hosts and organizers. And it's kind of tough because I've said some pretty mean things about evolutionary biologists in the past. And as a code of conduct, I have to kind of tone that down if I name names, even if I have a lot of hostility to them. There's a certain level of collegialness that is expected of me. And even if I never attend another conference in the future, I still feel like, as part of the reciprocity, I have to show a little bit of decorum in the future. And I have to set my feelings aside and say, hey, I have to set an example for other creationists.
Yeah. Well, I was surprised when you told me that you actually were invited to an evolution conference. And my first thought was, well, how'd you pull that off? Are you posing as an evolutionist?
Well,
because there's so much censorship, but so it's great that they did allow you to come on and give what turned out to be the opening presentation. And right now, the one that gets the most views.
That's right. So how did this happen? I was talking to Dan Stern Cardinale. He's the one, if if you've seen some other shows where he comes on and says, we both agree there's no universal common ancestor for all proteins. And I know that when I first put that on the table, it was almost hard to believe that I said it.
And he's an evolutionist. He's got a
yes,
fairly popular YouTube channel,
yes.
Dr. Dan Cardinal, and he's had you on several times.
And then so I posed these kind of questions to him, and he said, "You know what, Sal? You know what he said, what I don't like about what goes on your side. You never come to these conferences." He said, "Maybe you should go there and speak and just see the reaction." And I said, "Okay, I think that's a good idea. At least I could clear my conscience and say I tried. And if they canceled me, I could just report back. I say, hey, do you see how their conduct? This is why nothing is happening." So I decided to go. And I'm going to tell you a little backstory also. There are a lot of evolutionists that are switching sides.
Amen.
If not full blown, they're saying there's a problem. So in the last 20 years, this is like the 20-year anniversary of Kitzmiller versus Dover, and also the 20th anniversary of when I appeared on the cover of Nature. And people are really angry about that. A lot has changed since then. A lot has changed. So I was there with Stephen Meyer, Carolyn Crockers, Casey Luskin. And recently on the Joe Rogan Experience, I saw, and one of your guests, Emily Reeves, pointed me to this. And on that show, we had Brett Weinstein saying, modern Darwinism is broken. My colleagues, that is his Darwinist colleagues, are lying to themselves. They're lying to themselves. And ID proponent Stephen Meyer is a scientist who's actually quite good.
This is great. Great stuff. Modern Darwinism is broken.
And apparently some people are not getting the memo in the evolutionary community. So if you want to talk about climate change, this is a cultural climate change that I have sensed. And I personally know, and I felt I also needed to show up, if for no other reason, that I personally know of at least six evolutionary biologists or professors of evolutionary biology who have either, most of them are quietly, have switched sides.
Yeah.
And one of them was Richard Sternberg. Now, how could this possibly happen? Well, one of the cases I know of personally, this lady, when she was a young lady, her boyfriend died of an overdose. And then she went, when she had to clean up his room, she found all this pro-Darwin stuff. It was very dark. And she felt that Darwin took away the love of her life. And, that at least it began to change her view of what she was doing. She became an evolutionary biologist.
Was she an evolutionary biologist at that time?
I actually don't know all the details. She's a published evolutionary biologist, but somewhere along the way, the scientific evidence started to persuade her. She said, this is all wrong. So you have kind of the very personal experience there. I don't know that changed her mind. It's like, well, maybe that, you can't deny the science. But then she realized it wasn't science to begin with. And then she's been on a quiet mission, but she's under the radar. And I know a lot of stories like that. I was reading something on population genetics theory by an evolutionary biologist. I said, this is the best stuff I've ever seen. And then I met the guy and he's like, he's an ID proponent. So you have a lot of people that in the course of their career, they're like, they begin to see the light and then they're stuck. Yeah. And I think it's just reassuring to say, hey, you're not alone. You're not alone. I can't maybe help fix where you are now because you have a profession that you might have to try to figure out how to navigate. And maybe when you figure that out, you can help other people because there are more and more every year. And evidence of that is Brett Weinstein coming out and say, my colleagues are lying to themselves. And if that's his colleagues, how many more out there are like, yeah, we are lying to ourselves. We have to come to terms with the evidence. So there's been a climate change of sorts. It is cultural, not meteorological.
Yeah. So. Well, and with the internet and just so much information coming out now on creation and just the evidence for intelligent design. It wasn't that long ago. We had Dr. Aaron Shoulders on the show, He's a molecular biologist professor at Colorado State University. So more and more of people are either coming out of the closet or they've been out of the closet, and there's less fear because the evidence is just growing. We are really making headway. Really thankful for people like you. So I want to dive into what you got into at this evolution conference. But before we do that, what kind of reaction did you actually get afterwards? I'm kind of starting at the end and then working backwards. Did you feel like you had a lot of scolding eyes at you, or was it
Not at
friendlier
the
than you expected?
it was friendlier than I expected. There were two components. I really wanted to go to the in-person, but there were some issues that prevented me from going. And so I decided to just do the online. And so it wasn't that I didn't try. And the online makes it possible for people all around the world to appear. So there'll be people from Singapore and they don't have to fly all the way here and then just talk for 15 minutes and then fly back home.
Mm-hmm.
There were like 1,000 evolutionary biologists at the conference. 1,000. This was a month-long affair. And so they had to kind of just herd us and say, look, there's so many of us, we only allocate 13 minutes to talk, 12 minutes preferable, 13 minutes to talk, 2 minutes Q&A, and then you're gone. So there was no opportunity for anyone to react to anyone. So in that sense, I was saved by the bell. So there was a lot of negative reaction after the conference. There were YouTube videos made about what I had to say. But as far as interacting with the other people, they just said thank you and moved on to the next person.
All right.
And then I got that letter. You're welcome back next year.
Yeah. Cool. Well, let's see if they go through with that. And you'll go back too, won't you? You got so much great stuff to present.
That is if I have. I'd like to go back with some experimental stuff and stuff. Maybe if we might have peer-reviewed papers published by then. And I definitely would. And so that's the other thing that helped me get in there. I'm one of the few creationists that have published stuff critical of Darwinian evolution in secular peer review. Mm-hmm. I've also published on other things in the secular press, like at Oxford University Press, Springer Nature, some Federation of the Federation of American Societies for Experimental Biology. So when I posted that, and then I said my co-authors, one's a famous geneticist, John Sanford, another's an emeritus professor of mathematics, another is a professor of mathematics and population genetics, and an award-winning mathematician who won the highest award for the nation in Sweden, that's Alla Hoster. They're like, well, this would look kind of bad if we if we didn't allow some people of this caliber to present at this conference.
Bad idea.
I think that helped me get through the door. They saw the abstract ahead of time, and it was very anti-Darwinian. I put it in the most polite language, but I think ultimately it's God's grace that got me through the door.
Yeah, and like you've said, though, there's a lot of evolutionary biologists. They're still, I'm sure, committed, either atheists or agnostics, and they admit that the modern Darwinism, neo-Darwinism, is basically dead. They have to come up with other ideas, and they're trying to, but what else is there to have a naturalistic explanation? But so I'm curious, what did you present at the conference? So you said it, you had to jam this stuff into 13 minutes.
Oh my goodness.
We're not going to have a clock here and make you do that in 13 minutes. But
I said so it touches on things that I've said on the show, and one of the title of the topic was incorporating biophysical benchmarks into the notion of fitness. So we know that the notion of fitness in evolutionary biology has a lot of problems. Even Richard Lewontin said, it's not entirely clear what fitness is. And there's and this is a quote from him, I'm doing this from memory, he said, no concept in evolutionary biology has been more confusing than fitness. And he's saying it's not entirely clear. And then Andreas Wagner, who's also a very respected evolutionary biologist, said, to get an unassailable measurement of fitness does not in practice exist. It's hard to define. And I'm like, how can you do science if you cannot adequately define your central quantity?
Yeah, a
And if you
major
can't
variable.
measure it, can you do any science if you can't measure it?
Yeah.
This is a catastrophe.
And they base a lot of stuff off of, would this be population genetics and
A lot of
fitness?
population
Yeah.
genetics is based on fitness.
Yeah.
Now the math works out, but you have this thing called like W, capital W is like in some books absolute fitness and little w is relative fitness.
Mhm.
And all it measures is reproductive efficiency. That's all it is in a given context. And the problem is if you don't give the context, it's relatively meaningless. So all your equations, it's like, well, if you don't incorporate the context, it's going to be meaningless because you'll just say fitness goes up or down. But the thing of interest is how does it, going all the way back to Darwin, is how does this evolve organs of extreme perfection and complication? Origin of Species, Chapter 6. And so Darwin postulated that this process of getting the most reproductively efficient in one generation to be more represented in each in the process of what we call fixation. This is how you make organs of extreme perfection and complication. And I went first to great pains to show what are organs of extreme perfection and complication. And I have some examples here from the scientific literature. So they do exist. Now, what I want to point out is one reason I went through the trouble of showing this, there is a segment of the evolutionary community that's very uncomfortable with the idea that there is exquisite design in biology. That makes them uncomfortable. What would really make them uncomfortable is if those designs are better than all the scientists in the world put together. If they cannot make something that we see in biology, that would really make them uncomfortable. So,
So they must be super uncomfortable because we know.
yes, yes. I mean,
Now, were you presenting this type of argument?
I was presenting. I said, look, this is Eugene Koonin, who's the best evolutionary biologist in the planet. He said, biology is the new condensed matter physics. Biology is the new condensed matter physics. So we know that in Theodosius Dobzhansky said nothing in biology makes sense except in the light of evolution. That's wrong.
Wrong.
But it's now wrong on many levels because if biology is the new condensed matter physics, then nothing in biology makes sense except in the light of physics and engineering, because that's the best description of it. If you want to understand how biology operates, you don't need any more phylogenetic trees that are conflicted and probably useless anyway. What you need is you need engineers trying to figure out how the parts connect. First, identifying the parts, how the parts connect and why the parts are there, and what do they do, structure and function. And
it seems like to me we're seeing the word engineer.
Yes.
Engineered more often.
Yes.
Now, if something is engineered, doesn't that mean you need to have an engineer? I mean, it's
yes.
Bingo. They've got to, some of them have got to just cringe when other evolutionists use the word engineer. This thing was engineered.
Yes.
But we're seeing more of that.
And it's getting embedded. Okay, so from the National Institutes of Health and the Defense Advanced Research Project Agency and the National Academy of Science, it's forgotten. In 2006, they had this book, Computing in Biology, but it had a section in it that said, an engineering perspective on biological organisms. Biological organisms as engineered entities. Engineering insights can be useful in understanding biological organisms as engineered entities. And it goes far beyond that now, because as we have probed biological systems, we're seeing that it solves problems at the nanomolecular level and at the quantum realm that we don't know how to solve. And it's like biology is several orders of magnitude advanced technology. And I was just sharing that. I said, look, if you want a metric for what fit is, why don't we use biophysics to determine, now you have an objective standard to say, this is good, this is not quite as good. And so I was giving examples of biological systems that operate at the limits of physics. I said, that is as fit in the engineering sense that it's ever going to get. Nothing can do better. So why don't we use that as our metric? So if you want to try to maximize the fitness of something, now you have an objective benchmark to do that rather than this nonsense metric. I call this cow pie fitness. I said, well, I didn't use this at the conference. I was far too polite to actually say that. But I said, why are you using, in my head, I was saying, why are you using this cow pie metric? Why don't you use biophysical metrics? Now you can actually tell if you're reaching that through an evolutionary Darwinian process. And I argued that you can't. The evidence, the experimental evidence, first off, is showing that it doesn't agree. Darwinian process do not make this. And I actually used that little graph that I did when I said proteins are platonic forms, and that's why Dr. Dan Stern Cardinal said there's no common ancestor for all proteins. I said, look, a Darwinian process cannot reach this. It just kind of pops out of nowhere. I managed to sneak that one in, and
At the conference.
At the conference. Okay. I mean, that was kind of but I showed a peer-reviewed paper by Michael Denton that said proteins, the pre-Darwinian conception of proteins as platonic forms, and the role of physics. So
platonic forms. Just for our audience, you want to elaborate on that?
I actually don't know the definition, but it's kind of like it's just kind of like the idea you have a piston and you have a battery in a car. They're not the same thing. We see that a lot in engineering, that you could say a piston or a key or a lock, and engineers would understand, even ordinary people would understand, these are idealized objects that you can make. And so, if you say a battery, that has a certain construction to it, and we have all sorts of varieties, but underlying that is kind of a certain construction that has certain elements. And that's the way it is with proteins. You cannot evolve, like, say, one class of proteins, and we have motor proteins, to be other kinds of proteins, like structural. It just doesn't work. And we had a whole show on that.
Yeah.
And that, by the way, that's a great show to look at.
You have to have them from the beginning.
You have to have them from the beginning. And I threw that on the table, and I said, you know what? Darwinian processes do not, they are challenged to maintain them to begin with, because I showed all these experiments, like Lenski's, where it says genomes decay despite sustained fitness gains. And I said, look, it can't maintain this. And the reason is, by the way, I didn't have time to explain, it's metabolic efficiency. So way back in 1965, they had Spiegelman's experiment where they had an artificial, quote unquote, artificial life form. It really was just a string of RNAs, okay? Mm-hmm. It started out with several thousand, and after 74 generations, it was only 218 long. And everyone was saying, look, that's selection at work. This is Darwinian processes validated. Selection works. It evolved it. And I'm like, thinking to myself, you're actually celebrating this? You went from a long genome into a reduced one.
Yeah.
You have to go the other way.
Roasted.
So they're applauding basically de-evolution.
Yes.
They're celebrating something going backwards.
Going backwards. Now, before we had all the cheap genome sequencing, the price of genome sequencing has dropped 1 million times. So now any laboratory studying any organism, if they have even a moderate amount of money, they can send it off to get a gene sequenced. And so now they're counting the genes before something evolved. And they're like, well, gee, under extreme environmental pressure, the first thing to do is like dumping a backpack full of all your gear. If you don't immediately need it, dispose of it. So now what ends up happening is you're maladapted to work in other environments. And we call that loss of versatility. Mm-hmm. So what we had assumed is, oh, we're evolving something to be more of a specialist. But what happens when you're becoming more of a specialist? Yep. You become less of a generalist. And now you're maladapted to all these other environments. And we started to do more broad-based experiments. It's like, oh, it's quote unquote more cow pie fit in this environment. It's totally unfit in all these 100 others. So was that really an improvement? You've lost all this versatility. And this is being borne out by experiment after experiment. And I was just pointing that out. And it's like, you can't really give me pushback for just saying the facts. I mean, you might want to, but this is your own literature. So my strategy is I'm just going to quote your own literature. I quoted Lewontin saying your definition doesn't work. I quoted Wagner. You can't measure it. And the other thing is Darwinian processes destroy all these genes. So that's anti-correlation.
Yep.
How can you argue with that, you know?
So what was the highlight of your talk? Like, did you come to a crescendo on any particular topic? Because again, you had to jam a lot in 13 minutes.
Oh, goodness. So we have this Proceedings to National Academy of Sciences paper in 2017, and they're specifically addressing the backward wired retina. And this has been often used as by evolutionists to say, you know, the designer's incompetent.
Oh, you and I have heard that too. For years, we've heard that.
I know.
Bob actually interviewed a guy who wrote a book on the evolution of the eye.
Yeah.
And there wasn't one part in his entire book about how the eye evolved. You think if you're going to write a book about how the eye evolved, you have some evidence in there or even mention how it evolved.
Exactly. So here's this paper. It says, ingeniously designed, and I repeated that 4 times. I said, as an engineer, I love that phrase. Ingeniously designed. Ingeniously designed.
For something that for years they claimed was, backwards engineering and all this kind of.
And I didn't want to rub it in too much, but I said that if you take that acronym, that's ID. I said there's ID in biology. I didn't rub that in. But I said, for ID proponents who want to publish in peer review, you could just cite this paper. This is an ingenious design, and just put that little citation. So there you have it. Now, there are a lot of engineers that are still into the Darwinian paradigm, but they're at least saying this is ingenious. Mm-hmm. This is just amazing. And then I quoted Emmanuel Todorov, who is a professor of robotics. And in far back as 2010, he said, well, you might say the human body is sloppy, but no, we're better designed than any robot. You just can't get around that.
Far better designed. Yeah, not even close.
And that agrees with Stuart Burgess, who has numerous peer-reviewed papers, peer-reviewed by the community of robotics and biomimetics and bio-inspired designs and the Institute of Physics. So you can't run away from this. And
Stuart Burgess, is he a creationist?
He's a young earth creationist, but he is so respected. He's an editor of a secular peer. He's co-editor of secular peer-reviewed journal. He has made robots and all sorts of devices for spaceships and all sorts of things, like even the bicycle team for the UK, for Britain.
Wow.
I mean, he's a mechanical engineer. And I did cite this by Daniel Dennett. Biology is engineering. So you're seeing this. We can't run away from it because we have here, for example, this one, and I quoted this in my talk, some superior technological secrets have come to light from deep sea organisms. Modern technology cannot yet compete with some of the sophisticated optical systems possessed by biological organisms. Is that telling you that something engineered at the nano level is done better than anything we can do? Shouldn't that unsettle some people who are,
Yeah, you got to wonder if, so this is kind of towards the end of your talk, right?
No, I opened up with it. You asked me what was my favorite. It was actually.
Okay, so you actually opened up with this hard hitting.
Oh, yeah. I was just saying, look, this is good stuff. And I just kept going through all of this. I talked about the quantum compass. The quantum compass, the bird can navigate from Alaska, 14,000 kilometers all the way to Australia, point to point. And it uses a quantum compass. They figured this out. And there are various titles associated with this. Sustained quantum coherence and entanglement in the avian compass. And the way this is able to sustain it exceeds the durations achieved in the best comparable man-made molecular systems. We don't know how to do this. This is so much better.
So it says entanglement. Is that part of quantum entanglement?
Yes, it is. It's using quantum entanglement. It's using the entanglement to sense the spin of the electron. As the bird flies through the magnetic fields, it's altering the spin of the electron. It can sense that. We can't do anything like that. We can't do that. And I'll show you.
And that's instantaneous. So this to me, I'm going to say, hey, this looks like information traveling faster than the speed of light.
I
I'm just going to throw that out there,
yeah,
Sal.
yes, throw that out there by all means, definitely. And by the way, we talk about the limits of what physics will allow. It says here, biological magnetic sensing comes close to the quantum limit. We cannot even get that close. This is right at the limit. It's man-made devices are not going to get better. It's like there's a designer here. What made this design so good?
And this is just from this year, January 2025 from Physics.
Wow.
Magazine.
And this is an example, by the way, of a man-made attempt to make a comparable quantum compass. Now, this is an accelerometer. It has, if you wonder
For the
why.
radio audience, this thing is how big? This thing's probably what, about 4 feet tall, 4 feet by 4
Yeah,
feet?
you need a van. You need a van to be able to transport this baby. You just see kind of the core of the compass.
Something that is somewhere in a bird's brain.
Eye.
Here it is.
There it is.
And it looks terrible from our perspective as an engineer looking at this thing and then comparing that to a bird.
Yes.
It's like, okay, if I'm a customer of a product, I'm like, I think I'll take the bird version.
Yes. And see, the bird is able to operate at warm temperatures with lots of liquid around it. I mean, water liquid.
Yeah.
This, the man-made version, it needs to be, you need a cryogenic refrigerator to generate cryogenic liquid, and then it's going to be cooling down the system. And yeah,
Remarkable. Wow.
Yeah. How are you gonna put this inside any sort of? You're definitely not going to put it on like little small aerial drone. And the idea was they wanted to replace GPS, so the bird had something that could navigate as precisely as GPS even before we had GPS satellites. This is incredible. So I only had a chance to show some of these. And then there's a quote by Natalie Angier somewhere that I quoted, and I said, it's become fashionable in physics to say life is more perfect than we imagined. And this is a lecture that William Bialik makes. He's from Princeton University, National Academy of Sciences. And let's just look at some examples. The brain operates at 20, 25 watts, whereas if we tried to do this with silicon-based VLSI transistors, it'd take 10 million watts, 10 million. Yeah. So this is how efficient this, I speculated this is probably at the limit of physics. Next, biocatalysts. Enzymes are biocatalysts. They catalyze reactions. They are so efficient that we can't make anything from scratch that can operate this well. Like, for example, the topoisomerase, which I published in. We can't even build a topoisomerase that operates at any efficiency, much less something as efficient as this. Now, we can take these things out of a biological context and make them run faster, but that's really cheating. So these things are really well optimized. They're close to the limits of physics. Next, the shark can sense electric fields, probably at the limits of physics, because it can sense the electric fields of its prey. When the prey starts to move its muscles, it generates an electric field. The shark can sense it it's just incredible.
Yeah, we had a show not that long ago
right.
talking about the electric field that humans have.
Right.
So, a lot of people don't even think of that.
Exactly. And the eye can detect a single photon, the nose a single molecule.
So if you go scuba diving and wear a lot of rubber, the shark won't see you.
I doubt it. It'll see you. It'll see you. Just don't move too much because it can sense your electric fields moving your muscles. And so Natalie Angier, as she interviewed all these biophysicists, she concluded, as far as these systems go, she says, the system couldn't get faster, more sensitive, or more efficient without first relocating to an alternate universe with alternate physical constants. And so I was pointing out, we can objectively say, this is probably what helped me get in. I said, I have to give some credit to Darwin. Darwin used the term organs of extreme perfection and complication for biology. It's funny, the modern Darwinists, half of them want to say it's junky, it's garbage, it's bad. There's another half, like Richard Dawkins and Daniel Dennett, say, no, it's good. So they can't agree with each other. But I said, look, the physicists have affirmed Darwin's view that there are organs of extreme perfection and complication. So if if there's nothing else that I said, they ought to be able to walk away happy with that. We affirmed something Darwin actually said, and I did. I can, in good conscience as a creationist, say he was absolutely right. These are organs of extreme perfection and complication.
You know, I look at this quote and boy, they love to appeal to alternative universes, many worlds theory to get around serious.
Yes.
It's like it's in their subconscious that somehow she had to refer to this.
Yes. And so, yes, I'm sure it's in the back of her mind because Natalie Angier is a stark raving atheist. I think at the time she wrote this in 2010, she still believed in the Darwinian viewpoint. And it was still kind of a thing back then. It's continuing to decline now. There's some diehards who are still holding on to it. It's like holding on to a zombie. And so then the next part was, well, Darwin got this right, give him credit. But the problem is his theory says that Darwinian processes ought to maintain this. It can't even maintain it. So if you can't even maintain this, much less are you able to build it. That was kind of the logic. I mean, if you can't maintain something that's working, how much harder is it going to be to build it? So then I showed a whole bunch of slides. This is one of my peer-reviewed publications with John Sanford. We actually covered this, but we weren't saying anything new. And for example, Lenski, 2017, genomes decay despite sustained fitness gains. This means that these organs of extreme perfection and complication are decaying away, even though the supposed fitness is increasing. I said, there's got to be a problem with the way you define fitness.
And this was with bacteria, which bacteria
bacteria
reproduce rapidly. So it'd be a perfect thing to look at for evolution, right? But they don't see anything.
And then so I showed, they said, well, when you take this and you take these strains, try to test them in other environments, they utter fail. And it was Lenski himself who was involved in that study. So you could see that on the left panel, in that one restricted environment, the mean fitness, really the measure of reproductive efficiency, it kept increasing. So that's what it means. The fitness was increasing. But if you look at the effect of losing all those genes and you take that bacteria to like, say, 100 different other environments, you could tell it's just not coping well.
Yeah.
The mean fitness has declined. So I said, this is more the real picture. And this all obviously should make one think, well, what does it really represent? If you're only going to represent one environment and not capture its effect on others, isn't your definition of fitness kind of like at least complete?
Yep.
Pretty
fuzzy.
Yes, but I was pointing out this is a genetic decay, and I was arguing this is pervasive. This isn't an isolated environment and an isolated instance. I'm sorry, it's not an isolated instance. This is the rule. There may be a few exceptions, but this is the rule. You can't be citing exceptions to pretend that that's the rule. So
and again, you're using something that has rapid reproduction,
right?
Bacteria.
And I cited
even there, they can't get evolution. They get
right
decreasing, really decreasing ability to function in other environments when you're mutating it like that. Is that what you're saying, pretty much, or
that is exactly it? And then also, for the human genome, now it doesn't reproduce as quickly, but we're more complex. There's for something, and we know when we deal with more and more complex systems, there's more places for it to break. So there are many ways to break, few ways to make. And humans are so complicated that a bacteria could probably tolerate 80% change of its genome if it's in the right environment, lives. That's unimaginable for a human. Totally. I mean, even one gene could if it's wrong, could wipe out a human system. I mean, the human's dead. But for humans, I cited the fact that all studies are showing we're
decaying.
We are
Every
decaying.
generation
We are decaying over time.
Yeah.
I didn't want to rub it in, but I had a debate with Dr. Dan Stern Cardinal 5 years ago, and I gave a list of all these evolutionary biologists that express concern about the human genome decaying. And he said, no, that's an appeal to authority, Sal.
We're appealing to your authority.
And then I said, Dr. Dan, can you name one evolutionary biologist or geneticist of any repute that thinks the human genome's improving. He gave that like deer in the headlights look for 5 seconds, silence, and then he said no, and then changed the subject.
Oh man, that's classic.
So I pointed this out, not the whole conversation. I said, look, there's decay in the human genome, and I cited a study on, I think it's by Kawanaga, something like Kanazawa, I think is his name. I don't have that slide here, but it says intelligence and childlessness in women. Very intelligent women have a 28% higher incidence of childlessness. They have fewer kids, many of them are childless. And what does that mean? They said the implication is, so a Darwinian process is going to be getting rid of. I said, look, this is undeniable. We have experimental evidence. IQ tests are debatable, but we can measure nerve conduction speed, which is a proxy for intelligence. So as we get older, our conduction speed, sadly, starts to go down, or our mental faculties are not as sharp. Mm-hmm. So that's an objective measure. The nerve conduction.
That's interesting. I haven't heard that before.
Oh yeah, they wanted to have they measure
measurement of
nerve conduct. It's NCV, nerve conduction velocity.
Wow. Okay.
And so, well, you can kind of see it. Like, I mean, when I see baseball players that could be able to read pitches and react as they get older.
Okay, so that's part of the mechanism.
They're
never
not
thought of it like that.
The reflexes. Yeah, they said like the ping pong players who are like high caliber, after about age 25, the reflexes are just not quite there. Their ability to react. And I think that's related to nerve conduction speed. And they've been doing a lot of longitudinal studies on people as they age. And then also these studies cross-generational. It's like the nerve conduction speed has decayed. And so it's like, well,
so instead of saying I just had a senior moment, I had a nerve conduction velocity change.
You could.
Reduction.
I used to say I had a Joe Biden moment. So, I know that was mean. So I was pointing out, I said, look at all these examples. I said, well, this is, but I said, the bottom line is, all right, whatever you may think of, and I'm being quite a bit more verbose than I was in my presentation, but whatever you all may think of the hypothesis I put on the table, this is experimentally testable. You can test whether there's genomic decay in the human species and then for other species. And I did throw out a little slide. I said, we're in the middle of the 6th great extinction, according to, the consensus viewpoint. And you can't run away from that. We call, I didn't use this term, but I'll elaborate a little more. In statistics, we call this survivorship bias. If you only focus on the things that actually survived a process, you're kind of skewing the odds of how well your theory really applies. Yeah. Because if the environment changes and you have all these creatures that don't adapt, why don't you include that to talk about the efficacy of Darwinian processes? Because a lot of them are not able to adapt, lots of them. And then the ones that quote unquote do adapt, they're losing genes. The genome decays. There's a sustained fitness gain, which in many evolutionary papers they interpret as, oh, these creatures got more adapted. And I'm just like, yeah. I saw a peer-reviewed paper, by the way. It said gene loss is a key evolutionary force.
Yeah.
And that wasn't from The Onion. That was a real peer-reviewed paper. Oxford University Press.
Well, I've mentioned before on the show, I've had evolutionary biologists tell me, PhD scientists, that cancer is an example of evolution at work.
You gotta be kidding me.
A third grader could see how silly that is to use that as an argument for evolution. Of course, there's the whole malaria, sickle cell. But anyway, Sal, so you crammed all this stuff in 13 minutes. How did it end? I mean, were you kind of cut off, or do they have a timer?
I cut myself off. All the other presenters, all of us, because we were under a 15-minute hard time constraint. Yep. And they said, try to, stop talking at about 12, 13 minutes and leave 2 minutes for questions and setup. And so I cut myself off at 13 minutes.
Did you have questions?
One was like, can you give me citations for that? One young lady. And I was like, and I didn't realize that they didn't post my email. I said, you can email me, I'll give you a list. But I said, right now the best is in those slides. And
Yeah,
that's
the reason why I reacted that way is because, as you know, just years of debating evolutionists, if you say anything, where's the citation? Where's the peer reviewed? I mean, it's like a stock answer for them.
Who are your sources?
yes. And so like we did have a slideshow where I would just talk through it and you could actually see the papers, and she
Yeah.
was interested. And I'm just, and I have this, she's going to be in for a shock because over the next, I was just at a recent conference. I got to talk to a microbiologist, and after I spoke, I gave kind of a similar talk like this. He came in, he said, "Well, uh, Sal kind of took some of my thunder, but I have even more examples." And I asked him after he talked, I said, "How many?" He said, "I have like 50. I have 50 peer-reviewed papers." And I kind of went out on a limb with my few. Those are the best I could. You got 50 of them.
So I missed something here. So 50 peer-reviewed papers that are kind of pushing ID, intelligent design, or what did I miss here?
Decay of genomes.
Oh, decay of genomes. Okay,
And so
which might as well be, yeah. I mean, that's as brutally almost fatal to evolution as you can get, right?
they're going to be
I'm
more.
not being nice by saying almost fatal. It really is fatal.
And it's like, can you give me one example of a complex? And I specifically said, this is the requirement for, I said, there's all these transcripts that may be junk. So a cancer cell can easily generate new genes. All right. This is like taking a sledgehammer to a nice car and just beating it up and say, hey, we have a new car. It changed,
Yeah.
and I said, no, what I want, the measure of this would be, a new protein that has what we call a quaternary structure that its functioning depends on its multimeric quaternary structure. That's a fancy way of saying it, but that's a term they use in biochemistry. You need to have something that has multiple parts, at least two, and it's critically dependent on two parts because just like having a piston fit exactly in the engine block, the probability of two random parts being able to so well fit like a lock and key, that's very difficult. You can just claim any random cancerous transcript is a new gene. That doesn't count. You need something where you have interlocking parts, lock and key, and they would know that this is not easy. You do not get that. You don't get a random lock and key. You can just make up a key and say that's sort of functional, can kind of work like a hammer,
Yeah.
not very good. But to get it to work like something that opens a lock, that's very improbable. And so I threw that out there, and you could tell I crammed in a lot. And I know that would be a problem, and that was specifically what Dan Stern Cardinal was saying. That's a really good problem, Sal. Why don't you pose that to the audience and see how they react? Of course, as I said, there were so many presentations, I got drowned out in all the presentations.
Did you get any feedback on that one, either during the show or after, or the presentation, the conference, or later in email?
I haven't seen it. There've been shows that I just haven't had time to watch, and I'm not that interested. I can almost predict what they're going to say, and I actually had so many other things on my plate. I have like 3 peer-reviewed papers kind of in the pipeline with a deadline from an editor.
Okay.
So, and there's so many other things going on. And I just didn't have time to waste, because that's a testable prediction. I could almost write a critique of myself because I've been around them for 20 years. I could almost say, what they're going to say. So,
Yep. Okay. So do you have anything major in the works that you can share with us now? So moving past the conference, I know.
Well, I did talk about Exitons.
Oh, you were mentioning that off air.
Yes,
Those
I
sound
meant.
super interesting.
So I mentioned Exitons in the talk, and I just showed a slide. I said the Exiton transport is near 100%. I used the word 100% efficient, and Dr. Dan corrected. That was the one critique I did here. He said it can't be 100%. I said, okay, nearly 100%. And what an exciton is, this is the new frontier of biology, the biophysics at the nano quantum scale. So you saw with the quantum magnetic compass, that's one taste of it. The quantum entanglement. There is this thing called quasi-particles. quasi-particles, there is a new view in physics that all particles are quasi, and they kind of emerge out of, even possibly relativity comes out of the quantum foam. And there were uh, four Nobel Prizes shared among 11 physicists, at least, on quasi-particle theory. So when you look at a diode, for example, it takes an exciton and it annihilates it. The proper term is recombination. So when you look at a diode, a light-emitting diode, it uses excitons to make that light. Photosynthesis does the reverse. It takes light and makes an exciton particle. An exciton particle is a neutrally charged particle that has about twice the weight of an electron, because it's composed in part by a regular electron and then what they call an electron hole, which is a positively charged particle that has position, it has velocity, and it has mass. And it's called an electron hole. I wish they didn't call it that, because it's not really a hole, it's a real particle.
Mm-hmm.
And I mean, there's an argument whether they're virtual or real. And it's like, well, if you look at David Snowk's book, he's an ID proponent, he's a distinguished professor of physics, and he has a book from that he wrote that was published by Cambridge University Press. He is absolutely brilliant, and a lot of his solid state physics book deals with quasi-particles. It would be kind of like the Bible of quasi-particles. He has been an expert that has been able to create these Bose-Einstein condensate. For lack of a better word, and he's probably going to cringe if he's listening, a condensate's like, when if you run a distillery, you're creating condensates, right? You can take quasi-particles and make condensates. The condensate is made of these quasi-particles, and it's referred to as the fifth state of matter. And the way some speculate that how these excitons are able to transport, they go into these excitons go into a fifth state of matter. They're like a superfluid that's frictionless, and it can move through there. I said we didn't even know we could make excitons until recently,
Mm-hmm.
And much less a fluid made of excitons. And then we could do it at room temperature and get it to move through, through a plant photosynthesis system. And we have just scratched the surface. They are so full. Oh, here's a quote from University of Chicago News. The finding is scientifically intriguing and may suggest new ways to think about designing electronics. And when I pointed out at Evolution 2025, I said, don't write this off, because where do you think all the money and grants are going to go? Because engineers have these problems they're trying to solve in nano electronics, nano engineering, and it's already solved in biology.
Yeah.
And we need to do technology transfer. If you want to, you could either, I didn't say this, but the implication is you can either be run over by the train or you can try to hop on board. Mm-hmm. There's a lot of experimental evolution.
Now, what's the engineering benefit of this?
If you can design better solar cells.
Solar cells.
So solar cells have had efficiency ratings of like 5% and 10. I think they're up. Some of the better ones are, I've heard numbers like 27%, but there's none that's 100%. None that are. Now, even photosynthesis isn't 100%, but one component, which is the exciton transport, is. And so also, if we can, this isn't exactly superconductivity, but if you can pump electrical things that have electrical energy in them, like the excitons, if you can pump it through a system without any friction, you almost as good if you can push it through a wire. And also in digital communications, if you can get excitons to move through, they are immune to Ohm's law.
Okay.
So you get all these cool particles. Yeah. And if you look on Wikipedia, list of quasi-particles, you'll see it's a gigantic list. We're discovering more every day.
And I know, the power grid is under a lot of stress. I have a brother-in-law who's an engineer for Xcel, which, is a power company that powers Colorado and I think several adjacent states. This could be something that helps relieve that huge strain that's on the power grid right now.
Right.
Worse than I think people realize.
and so now this is like the engineers. I'm just like, the engineers have an opportunity now to start to build even cooler toys. And I say, if you want to engineer cool toys, study biology, because that's where the coolest action is. You'll see design at its best.
Absolutely. And the more we can reverse engineer, the better life's going to be for everybody.
Which means also there are going to be more engineers involved in biology,
Oh, yeah.
more physicists. The
Yeah.
last thing we need is guys just making more phylogenetic trees. And
Yeah.
I do want to make one comment. A lot of the people there at the conference are very lovely people. If we didn't have such opposing metaphysical views, they had the kind of personality that you'd want to work in a lab with them. You'd want to hang out with them at the restaurant or at the bar even, or wherever. They're just very sweet, humble people. They're not like the people you do see on the internet who are what I call evolutionary propagandists, like Richard Dawkins, and who are so hostile to Christians and so forth.
Yep.
Most of them by and large are very meek. They're quiet. They just want to be in their lab and they want to be writing papers and doing experiments. So, it was eye-opening to me to see that, there's a level of, there's a level of hostility, in the public sphere that's not actually there at the personal level for many of them. There's still, people like Richard Dawkins and Nathan Lentz, and then all of, Dave Farina. Oh my goodness.
Yeah. Yep.
That doesn't represent the evolutionary community. And I'm glad to say many of them are starting to become like Brett Weinstein. It's like, hey guys, you're lying to yourselves. You're lying to yourselves.
Sal, you're doing a lot of great work, and
Thank you.
We're looking forward to more research. I know you're on the cutting edge. You're our Real Science Radio's cutting edge guy.
Yeah, so I'm glad to have had the chance to introduce quasi-particles.
Yep.
That
Well, we'll have you on again.
Oh yeah,
We
definitely.
gotta, dive into that a little bit more, because we really value this kind of new research. I learned a lot of stuff. I always do on Real Science Radio, especially when you come on. But
Thanks.
this whole the technology that could improve solar cells. And sorry, Sal, it's Exitrons.
Exiton? I don't even know that I'm pronouncing it correct. Excitons, but there are other kinds of quasi-particles like polarons and Cooper pairs, which enable superconducting. There's a lot of research in using DNA to make superconductors. I mean, this is. I was just like, I don't think the evolutionary community realizes. I mean, I was the first to kind of land the beachhead. I said, you can ignore me. You're not going to ignore what you're going to see when you start seeing all these labs, and if you don't jump on this gravy train, you're going to be bypassed. You're going to miss the train. You're going to see all this advancement in technology. And if all you can do is just make more phylogenetic trees with your coloring books, you're going to be, I mean, that may sound harsh, but I think I felt my heart go out to them. I said, these are lovely people. They're dedicated. They're educated. They just need to evolve themselves and their viewpoint, and they can participate in this great enterprise. We're going to discover all the great designs that God has made, and you can appreciate it. You can help use it to further society. I mean, stop playing with your coloring books. I know that's so condescending, but you have these fine minds, and it's like there used to be a saying, it's a sad thing to put a mind to waste. And that's what I'm seeing. I said, these lovely people with intelligence, and it was my chance to give an outreach. And I am glad to say, like I said, there are evolutionary biologists I know who've quietly, at least in their heart, have switched sides. Some of them have left their profession as evolutionary biologists. And, it's a hard transition to make.
Yeah.
So.
Yeah, it's been part of your life. So, God's wonderful creation. Is, what's the Psalm? He has his wonderful things were made to be remembered. I'm paraphrasing. But, Sal, thanks again for coming on Real Science Radio. It's always great to have you. And, man, it's great to have you in studio.
Oh, yes.
And, next time you're, traveling the country, you driving across the United States of America, you stop into Real Science Radio studio. here in Broomfield, Colorado.
And I'll be better practiced to play on your lovely piano.
Oh yeah. Oh man. Yeah, Sal got to play. What was it you were playing? I know it's classical music.
Liebestrom by Franz Liszt.
Wow. He did a fantastic job. It was great. Yeah. My wife kind of, well, what's going on in there? She comes in and she's like, wow, that's really good. She sat down right on the couch and just chilled back and enjoyed some really good piano. So, anyways, yeah, you're quite the you have quite the talent there, Sal, in many ways. So, and it's good to have that artistic part of the brain too. So we're looking forward to you applying that too to continue the
Thank you.
creation research that you're doing. So, all right, well, I think we're out of time, Sal. So again, it's great to have you. So for Sal Cordova, I'm Fred Williams of Real Science Radio. May God bless you.